Hi, this is Sydney Hooker, and this is my tutorial on chapter 5.3, which is all about photoreception. In this outline that I wrote out, we're going to be first focusing on rods versus cones, which we're going to first focus on the anatomical and then move on to the functional differences between the two. And then the last three, we're going to be focusing on how researchers examine photoreceptive ability of an organism. And we got butterflies, birds, and snakes. And so we get to see through these three examples how they do that. We're going to be starting with part one of our outline, which focuses on photoreceptors, specifically rods versus cones, which is a very important distinction, especially as distinguished in this chapter. So, first of all, photoreceptors, as defined in our book, are specialized neurons that are sensitive to light and function as visual receptors. So, multiple wavelengths in the form of electromagnetic radiation are being bounced off different things in the environment, and our photoreceptors are what process that and turn it into visual stimulus for the brain. So, as you can see, this is a diagram of what I just explained. Light that's been bounced off other things in the environment gets reflected off into the eye through the lens and hits the back of the retina. The retina is where our photoreceptors are present. So we have two main types of photoreceptors. We have rods and we have cones. And there's more rods than cones within the eye in of itself. But the important distinguishments that the chapter wants you to focus on are rods are sensitive to a wide spectrum of light and they're best at low levels of illumination. Which means if you are looking for something in your dark room and you can't find it, this is where the rods are going to be the most helpful. They're going to be able to help you distinguish between different things, even if there's not a lot of bright light. But it sacrifices, since it has such a wide spectrum of sensitivity to light, it sacrifices color. So it has no color perception at all, just visual stimulus or black and white stimulus. When we go into cones, so they're sensitive to a lower range of light wavelengths, but that means they focus on three specific ones, particularly red, blue, and green wavelengths. And this allows us to see visible color. And it works best in, of course, bright light or, just going outside and you see the green trees. So yeah, big distinguishers there. The differences between rods and cones is just going to have to be memorized. But if you want a simple mnemonic, one way is you could focus on cones and realize it starts with a C, and then distinguish it as color vision, right? Rods doesn't really have a simpler way for that. There's not as clear of a mnemonic, but are going to have to memorize the difference there. So, as we moved on to the second part of our outline, we're going to observe how different researchers examine photoreceptive ability in different organisms. So we're going to start off with the Douglas Blackiston example, which has to do with monarch butterflies. So monarch butterflies already contain three opsin, ultraviolet, blue, and red and orange wavelengths, which means they could detect colored vision or colored light. But this does not mean it was true color vision. What Blackiston wanted to decide is to see if these monarch butterflies could determine, perceive, and discriminate amongst these wavelengths. So him and a couple of colleagues trained multiple monarch butterflies into associating a specific color, whether it be red, purple, blue, or yellow, and to perceive that with a food reward. So he trained these individual monarch butterflies in order to interpret their color with the food reward. After training them for a long time, they go ahead and present each of these butterflies with flowers. 20 flowers in total, 19 of which were a gray pigmentation, while one of them was the distinguished color that they were trained on. So in my example that I drew here, I did a couple gray flowers and one purple out of the four colors. Then they observed the monarch butterflies and see how much they probed or spent time with each of these flowers. And by the end of the study, the researchers determined that these butterflies were able to distinguish between different wavelengths of color and discriminate because 95% of the time they were on their trained distinguish color. And that demonstrated the butterflies' capabilities of true color vision. So we're going to be looking at UV detection using birds, and this is based off Satari's research with pied flycatchers. So they collected groups of male and female birds near their reproductive mating time, and they separated the males into two distinct groups. One group was control, and so they put a water-based substance on their feathers, while the experimental group, they put a UV-reduced sunscreen on their feathers. And they were separated. The males were not allowed to interact with one another, but the females were allowed to interact with each other. And then they opened it up to these certain nest points and observed which of the two groups did the female birds prefer to mate with. So, using this, we were able to see that the control, usually this is how the plumage would look. It would have a lighter variation on the top of the head with its normal distinction of colors in its normal way. While the UV reduced group, using the UV sunscreen, had a darker plumage on the top of its head. When looking at the final results of the study, 11 out of the 13 females preferred to be with the control over the UV-reduced males. So through this study, Satari determined that female birds assess male quality based on UV plumage coloration. So they don't only just perceive UV, but they also discriminate between it. For the final part of this presentation, we're going to be looking at snakes and their capabilities and reaction to infrared stimulus. So, cordyline snakes in particular have specific pit organs that allow them to detect infrared wavelengths. Infrared wavelengths are on the longer side in terms of like that instead of more like this, and so they are not in the visible wavelength spectrum for us humans. But with these specific adaptations like pit organs, it allows them to sense this type of wavelength. So in this experiment, Michael Grace and Adam Safer explored the differences between cordyline snakes that had these pit organs and viper snakes that did not. And they analyzed this by having these animals react to different balloons of different heats. So whether they be on the hotter side or on the cooler side, the researchers would then react to the snake's response to these different thermal temperature balloons. So the actions they were looking out for were tongue flicks, attacking the balloon, tail flicks. Any of these actions were seen as affirmative actions in this case. And so when they compared in the final graph between cordyline and viper snakes, they noticed that the cordyline snakes were more likely to have a higher tongue flick rate as well as other actions than the viper snake when in the presence of a warm target. This demonstrates how these snakes react to warm prey and would most likely attack or have a higher response rate to more warm targets. And this demonstrates how not only they can sense this difference, but also differentiate between the cold and the hot.